Human DNA is a double-stranded helix structure composed of nucleotides. Each nucleotide contains a sugar molecule, a phosphate group, and one of four nitrogenous bases, adenine, guanine, cytosine, or thymine. The human genome is made up of approximately 3 billion base pairs of DNA. More than 99% of human DNA is identical among all people. The remaining 1% of human DNA contains genetic variations that make us unique. DNA is organized into chromosomes, with humans having 23 pairs of chromosomes. Each chromosome contains many genes, which are segments of DNA that code for specific proteins. DNA is responsible for transmitting hereditary information from parents to offspring. Mutations in DNA can cause genetic disorders and diseases. DNA testing can be used for various purposes, such as identifying biological relatives, determining genetic predispositions to certain diseases, and forensic investigations. Recent advances in DNA sequencing technology have made it possible to study human genetic variation and to develop new treatments for genetic diseases. DNA replication is a process that occurs when a cell divides, resulting in two identical copies of DNA. DNA can be damaged by various factors, such as exposure to radiation, chemicals, and environmental toxins. The field of epigenetic studies changes in gene expression that are not caused by alterations in DNA sequence, but rather by modifications to the DNA molecule or its associated proteins. Mitochondrial DNA, which is inherited solely from the mother, is a small, circular piece of DNA that codes for proteins involved in energy production. DNA can be used as a storage medium for digital data, with scientists successfully encoding books, images, and even music in DNA molecules. Human DNA is highly similar to the DNA of other primates, such as chimpanzees and gorillas, indicating a shared evolutionary history. Genetic testing can be used to determine ancestry, providing insight into a person's ethnic background and migration patterns of their ancestors. Some people have genetic variations that give them resistance to certain diseases, such as sickle cell anemia and HIV. CRISPR-Cas9 is a revolutionary gene editing technology that allows scientists to make precise changes to DNA, opening up new possibilities for treating genetic disorders and diseases. The human genome is not static but can change over time due to natural selection, genetic drift, and gene flow. Junk DNA, also known as non-coding DNA, makes up a significant portion of the human genome and was once thought to be useless. However, recent research has revealed that some non-coding DNA has important functions, such as regulating gene expression. Telomeres are protective caps at the end of chromosomes that shorten as we age. Shortened telomeres are associated with various age-related diseases and may contribute to the aging process. Genetic testing can also be used to identify carrier status for certain genetic disorders, which can help individuals make informed decisions about family planning. DNA evidence is frequently used in criminal investigations and can be collected from various sources, such as blood, saliva, and hair. In addition to humans, DNA is found in all living organisms and provides the blueprint for their physical traits and behaviors. DNA sequencing technology has become faster, more accurate, and less expensive over the years, leading to new discoveries in genetics and personalized medicine. Genetic counseling is a specialized medical field that helps individuals and families understand and cope with the risk of inherited genetic disorders. The Human Cell Atlas is an international effort to map all the cells in the human body and understand how they interact with each other, with the goal of advancing our understanding of human biology and disease. Human DNA is incredibly complex, and much of its functionality and significance is still being discovered by scientists.